So welcome back. Application of PCR. Talking about traditional PCR, not quantitative PCR. So in traditional PCR, we have gone through its various mechanism, techniques, steps by step. Now the application, the way it's used is different. In cloning, we can amplify our large size of DNA, but in PCR, it's very difficult to amplify those huge size of DNA by this technique. So what is the sole application? What is the sole purpose to use this technique? Most of the students or even teachers don't used to tell this application to student, or even though they tell, they are not able to properly let the student understand. So I hope I could better try to let you understand that what is the sole purpose of this PCR, because it is a very small fragment of DNA can be amplified. So what is the reason that we are using in every single lab this technique? So here I am just going to discuss about the example. So whatever written here, forget about this. Let me just tell you, if you wish you can read or just try to understand what I am saying. So here two samples are taken. Cell here actually a hypothesis was made that we have a cell, and with increasing the glucose level, it increases the transcription of your endothelin 1 gene in microvascular endothelial cell. So microvascular endothelial cell is a cell which produce a type of glucose digesting or something gene, that is your endothelin 1 gene. So that gene which produced code for mRNA, and that mRNA will form a complementary DNA by reverse transcriptase. So we have to see that cDNA is expressed, and how we will compare that. Yes, in the glucose rich sample or solution, our endothelial cell, which is culture, shows overexpression of mRNA, or the gene is overexpressed because of this huge amount of or more amount of glucose than the normal glucose concentration of sample. So this endothelin 1 gene is the gene which is produced or expressed differently in different sample. Our first sample is your normal sample having no glucose or little amount of glucose, whereas our second sample possesses huge or more amount of glucose sample. So does this over amount of glucose affecting those endothelial cell to produce or to overexpress its gene to produce more number of DNA? And that as presumption or hypothesis we have made. To experimentally prove, we have to check the expression, the amount of cDNA that we have amplified, and we will compare by doing electrophoresis to check whether our DNA is really, the gene is really overexpressed with this environmental factor. So this is the thing. Hopefully you understand. So here are the things written here, everything. I'm not going to read all those things, you can read out. I'm just pausing it, have a screenshot or a picture, read out all those things that underlined or marked, but it's written everything I have told right now. So here our tube of cDNA will contain many sequences. So now we have cultured the cells in different solution having glucose rich and glucose poor. Now we have to take cell from this culture. Now during this taking of this cell, we may do some error. We'll discuss about that error later. Okay. Now while pipetting those cells, we have to take very few amount of cells, 1 to 2 up to 6 cells. So these cells containing those mRNA we have to be isolate, then we have to undergo this kind of things. But how we will isolate that particular endothelial 1 gene produced mRNA? That's not possible. So we have to take all those mRNA which is produced, we have to reverse transcriptase all those mRNA to make complementary DNA. Now the thing is important, because we have designed our primer in such a way that it will only complementary bind with that particular endothelial 1 gene mRNA, endothelial 1 gene cDNA. So when we run PCR, only that gene will be amplified, remaining all other cDNA will remain as such. Okay, so when we do this PCR, we will have to take two cell sample from the glucose pore and glucose rich sample. We have to add same amount of DNTP, same amount of enzymes, same amount of primer in both the tube, and that is very important, okay, to prevent any error. And after that, what we do, we will run it in PCR, and after running PCR we will have our desired DNA gene fragment, desired cDNA replicated, only those which we have interested in. Other cDNA will remain as such, they will not undergo any copy or replication, because we have used the primer. And even though DNA polymer is there, without primer DNA will not replicate. So we have designed a primer to complement it to that of our desired gene, so only those cDNA will be only replicating here. Okay, so after this we will get this kind of thing before doubling, and after doubling we can see a particular, that black dot is our desired endothelial 1 gene cDNA that is amplified up to this much. And now after that, this is then we have to isolate those cDNA and run it in electrophoresis. And in electrophoresis we have run the two sample of cDNA. You can see here it is normal and it is a high glucose. This two band showing the endothelial 1 gene cDNA is amplified, but in high glucose containing cell, the cDNA is which is copied is more as compared to your normal type of cell having that cDNA. So these two band look similar, but we cannot detect with our naked eyes, so we have to use computer program. By taking this pixel from 0 to 255, the computer can detect the difference between these two strands with their density, their compacting, and it can tell how much time this particular gene is present in our respective electrophoresis band or strip of electrophoresis. But due to some error, if we have made some instrumental error, or we have taken more sample, more number of cells, then there will be that much time of our increase in fold. So even though there will some error, this result, this error will not that much affect its result. So we can make sure, that the high glucose content cell has overexpression of your endothelial 1 gene, but it is your type of qualitative data. Quantitatively it's proved, because there must be some error. To prevent those kind of errors, we use a sample or a what we call sample gene. For example, some gene inside our cell has inside our cell continuously expressed, they are being required by every single time by this cell. So the mRNA produced in both the sample cell, that means from the poor glucose and rich glucose solution cells, both express a particular gene equally, even though they are placed in different environmental conditions. that kind of gene is your homeotic gene. Getting my point? So homeotic genes are the genes which express equally in different conditions because they are continuously required by the cell, not affected by these factors like glucose rich, glucose less. So let me show you this picture, you better understand. So in this case you can see here in both the sample we have taken cells, and according to our hypothesis this 18S rRNA is a type of gene, this is type of rRNA which we can also homeotic gene also here. Okay, so 18S rRNA must be expressed equally in both the cells. If we have make any error during pipetting, that's why it comes as 9 and 3. If there is no error in pipetting during pipetting, then there will be only 3 to 3 or 9 to 9 in both the cells. As it's showing, the amount of 18S RNA is high in this normal region. That means we have done some error while pipetting the number of cells from this sample. So to neutralize, to balance this thing, we can make balance by this way, by multiplying 3 with your high glucose or by dividing by 3 in your normal sample for that 18S rRNA. By doing that, we can see that there is 3 times factor that we have error. So to neutralize the endothelin 1 gene, we have to do the same thing. So in normal cell we divide by 3, or in high glucose we have to multiply with 3. That will show that this is the thing. So if we just do that, because it looks like both have the same amount of cDNA being expressed, that means even though they placed in different glucose level, the endothelial 1 gene is evenly expressed. That's not true, because we have some error, and this is clarified by this 18S rRNA. So now we have to neutralize this thing. And how we neutralize? It is a formula. You can read out all those things if you want. So this is the 3-fold difference. It is the formula, and we can see there's 3,000 folds. So actually in high glucose containing sample we have overexpression of your endothelial 1 gene. It is proved now, because 6 into 3 is equal to 18. So 18 times more mRNA is expressed, gene is overexpressed in this high glucose sample solution having culture of this endothelial cell. So this is the thing. So this is how we use this PCR technique to check whether during this particular condition our desired gene is being overexpressed or not, without having an error, by taking this homeotic gene or 18S rRNA as a checking indicator or indicator that relative strength. Here also some beta actin which also being expressed, some glycerol 3 phosphate dehydrogenase enzyme, other is like 18S ribosome RNA, etc. are being controlly used as an indicator. Okay, this is the thing. Next video will be qPCR. And why the quantitative PCR? Because in this traditional PCR we don't know that how much cycle is already run for a different strength. It's a little bit difficult to exactly know the quantitatively know, but the qPCR technique helps to overcome this problem also some extent. So that's what we'll discuss in next video. Thank you very much.